Okay, good afternoon, everyone, and welcome to today's UC Ag Expert Talk. I'm Stephanie Pereira with the UC Statewide IPM program. Cheryl Reynolds is also here with us to help troubleshoot any technical problems. Please note that this webinar is designed for growers and agricultural pest management professionals. Master gardeners can certainly benefit from participating, but the pest management methods presented, especially the pesticides, are not to be followed without a clear understanding of their legal use in home environments. Okay, so with that, I'd like to introduce our speaker today, Dr. Whitney Brim-DeForest. Dr. Brim-DeForest is the County Director for UC Cooperative Extension in Sutter and Yuba counties, as well as a Cooperative Extension Rice Advisor for Sutter, Yuba, Placer, and Sacramento counties. And today she'll be talking about weed management in rice. So now I'd like to pass this over to Whitney. Whitney, you can go ahead and share your slides.
Okay, thank you, Stephanie. Thank you for the introduction. Okay, well, it's great to see everyone here today. Thank you all for attending. I see some familiar names in the chat or in the participant list there, so thanks everyone again. All right, so I'm going to be speaking about weed management in California rice, and I'm going to try to do my best to kind of outline weed management in rice for folks that aren't familiar with rice before we get going into the research part of this. So weeds in California rice, we have many species, and what kind of makes rice unique in comparison to other cropping systems is that we have both aquatic weeds and those also adapted to drier conditions. Our weeds are very well adapted to our flooded rice culture. Most fields have been continuously flooded for roughly 100 years, and we normally continuously flood our rice fields even to this day. We originally actually started flooding for suppressing certain weed species, specifically barnyard grass. And barnyard grass is a weed in other cropping systems, but it is not well tolerant of flooded systems. We also have very little crop rotation or fallowing, except for this year, for example. This year we did have a considerable amount of fallowing, but normally we don't fallow very much, and we probably maybe roughly 10% of us or less do crop rotation on a normal basis. So therefore, our weeds are really managed through both flooding as well as herbicides. We don't have many other cultural controls that we're able to use. We have some unique herbicide applications as well. We apply both foliarly, like with a spray boom, and also into the water as a granular formulation, and that is pretty unique to rice. With no weed control, our losses in drill seeded systems, so that would be drill or dry seeded systems, can be up to 100%. And again, this emphasizes why we flood. Our losses in water seeded systems are closer to about 50%, roughly 50–60%. So you can see that just with that flooding, we're getting a really high level of weed control. So again, this is why we continuously flood. But as you see, or we'll see soon, it does pose some problems as well, this continuously flooded rice culture. I'll go quickly through some of our main weed species. So we really have just a few grass species. We have the Echinocloa complex, or the water grasses, as they're known commonly. We have Echinocloa cruscali, as I said earlier, which is barnyard grass. So that's the one that really, oops, my apologies, the one why we, which got us started flooding our rice fields in California. We also have late water grass, or Echinocloa philopogon, Echinocloa oryzoides, which is known as early water grass. And those two are both imports from Japan when we started farming in California in the early 1900s. And then the last one is Leptocloa fusca, which is a native species, and we actually have two subspecies of this one, commonly known as sprangletop. Our sedges and broadleaves, like I said before, we have the unique situation of having both aquatic weeds and non-aquatic weeds. So we have a considerable number of sedges. Sapirus deformis, also known as small flower umbrella sedge, which is very common worldwide in rice systems. It's not unique to California. Schunoplectus mucronatus, which is also known as rice-filled bulrush or rough seed, and that one is a native species. Amania, also known as red stem or breadberry. That's one of our broadleaves, and then we've got a couple other broadleaves. I don't have a picture of duck on here, I should. But duck is a common one. Sagittaria montevidensis is our arrowhead, one of our arrowhead species, and Elisma plantago aquatica, which I'm going to forget the common name for right now. But we have quite a few broadleaves. I'm not going to cover them in quite as great of detail today, mostly because they don't cause quite as much yield loss as our grasses. But suffice it to say, we have a bunch of different weed species, and they're each adapted to different systems of irrigation, which makes it rather difficult to manage. We also have some newer weed problems that have popped up in the past 10 years or so. I will be talking more about weedy rice later on, but weedy rice, suffice it to say, is a very common pest of rice worldwide. It is the same species as rice, so they're both a Rhiza sativa, and that makes it very hard to manage. So it is rice infesting rice, and as you can tell, that would be difficult both from the standpoint of recognizing it and also from the standpoint of control. So if we have herbicides that are registered to control weedy rice, the herbicides would also kill or control our rice varieties. So it makes it very difficult to manage. We have 6 confirmed biotypes in California, up from thinking that we had basically nothing about 10 years ago or so. And I'll talk about this a little bit more later on. So we have weedy rice, and then we also have this unknown species or biotype that is related to the ones that I spoke about earlier, the Panicles. So it's in the same family, and right now we're working on identifying it, and I'll speak about it a little bit more later. But what's making this so complicated in rice is that we've had several introductions of new species or new biotypes of different weeds in the past few years, and then we're also dealing with increasing regulations and then herbicide resistance, which I'll speak about in a moment. Our rice herbicides, we have not that many compared to other parts of the world, and also really compared to other cropping systems. I've got the common names listed here on the left. In the middle are the trade names, and on the right are the modes of action. So the thing that makes it really complicated for us currently is that we don't have that many modes of action. We have one new one introduced a couple of years ago, which is Butte, which is an HPPD inhibitor, sorry. And then we have primarily ALS inhibitors. We've got a couple other modes of action. Really, though, just very few in total. I don't have Prowl highlighted here because it's not useful in a flooded system. It can only be used in a dry seeded system. So really the ones that I've circled here are the ones that we have available for flooded systems. And I will go into our herbicide resistance issue here in a moment, but this makes it hard even from the standpoint of being able to rotate modes of action. Not all of these herbicides control all of our weeds that I mentioned previously. This has a lot of text on it, and I do apologize, but rice, California rice, holds the award for being or having the most number of weed species resistant to herbicides in the United States. So we have the most number, and we found our first in 1993. And as you can see, a lot of these are resistant to ALS inhibitors. A lot of herbicide resistance worldwide is resistant to or have species that are resistant to ALS inhibitors. But we also have added multiple resistance to many of our weed species. So you can see Echinocloa, late watergrass here. As of 2000, we have multiple resistance to 2 sites of action, ACCase inhibitors and lipid inhibitors. So we have many weed species that are resistant, and then we also have many that are multiple resistant. So they're multiple resistant to more than one mode of action in one species or biotype. So it's making it quite complicated to manage weeds in rice. So to give an idea of kind of where we're at here, for example, early watergrass, we have 8 registered herbicides. Those cover 6 modes of action, and we have herbicide resistance recorded in 4 modes of action, including some of those that have multiple resistance. I won't go through all of these, but Spangletop is another one that we're having issues controlling. So we have not that many herbicides registered to control Spangletop, so only 4 total, with 3 modes of action represented. And we already have 2 modes of action that we have recorded resistance in California. But in this case, at least, it's no multiple resistance. So they're only resistant to one herbicide and not the other. Okay, so I'm going to go quickly through known herbicide resistance here. But this will kind of walk us through the situation that we have going on here. So for PCAs and growers, this will hopefully kind of illustrate what we're dealing with here. So I mentioned earlier, right, that we have granular herbicides and foliar applied herbicides, and I've divided them into the 2 because usually these are applied at different times of the season. So for Spangletop, for example, we have granular herbicides. Each color represents a different mode of action. So Bolero, Serrano, Butte are 3 different modes of action, and Abolish and Bolero are the same mode of action. And Clincher is a different mode of action as well. So if you were going to plan a program for the year, this is kind of how I would sit down and go through my planning. So we have, like I mentioned before, known resistance to a couple of modes of action. One of those is Serrano or clomazone. So if you have Serrano resistance, what are you going to rotate with to set up your program? Rotate with Bolero or Butte, right? But then you only have 2 things to rotate with at the beginning of the season. And to get another mode of action in there, right, to help us prevent resistance or selection for resistance, you can't follow Bolero with Abolish because that's the same mode of action. So you need to follow Bolero with Clincher. Okay, so it begins to be quite complicated depending on if you already have known resistance in your field. A similar situation if you have Clincher resistance. So you'd have quite a bit to choose from at the beginning of the season, but as a follow-up foliar to catch your escapes, you've really only got Abolish. So that means you can't really put down Bolero at the beginning of the season. So you're left with Serrano and Butte. So you can tell it starts to get kind of complicated to plan your program. Small flower umbrella sedge is worse in a lot of ways here. So we have widespread resistance to the ALS inhibitors. So that takes out all of these herbicides. And as I mentioned before, a lot of our herbicides are ALS or in the mode of action of ALS inhibitors. So planning a program, right? Shark here and Shark here, you couldn't do that. Again, you couldn't do Bolero and Abolish in the same season. So again, it starts to get kind of complicated. So we have known ALS inhibitor resistance, and we've had it for a while. But then we recently added propanil resistance within the past like 5 or 6 years or so. So we have biotypes that are multiple resistant to both ALS inhibitors and propanil. So again, making it very complicated to plan a program that controls small flower umbrella sedge. And the reason, I'll just add really quickly, the reason that we got propanil resistance is because we tend to have a plan usually where we plan out our season to have granular at the beginning of the season and then follow up with propanil. And when you do that, you basically select for all the plants here at the beginning of the season that have resistance to propanil or to ALS inhibitors. And then those ones that escape, you're eventually going to start selecting for the ones that have resistance to both ALS and propanil, unless you rotate at the end of the season. So if you rotated with Shark one year, that would break that cycle of selecting for those resistant to propanil. Okay, I'm not going to go through all of these because they start to get a little bit similar here and repetitive, but rice field bulrush, again, known resistance. So ALS inhibitor resistance, and we did the same thing with small flower and bulrush. We added propanol resistance, both of them. Red stem is one of our only broadleaf weeds that actually we have resistance or known resistance in. So again, that ALS inhibitor resistance. Thankfully, this is not widespread, at least not that I know of. And then the last one is really the watergrass complex, and I've put these all in one category because they're all controlled by the same herbicides. Watergrass can also be known as mimic. So mimic would be the same species as late watergrass. And this is where things get a little bit complicated. So watergrass has what we call metabolic resistance, and that means that it's basically breaking down the herbicide. The plants are able to metabolize the herbicide. So we had originally the first few biotypes that were found with resistance to ALS inhibitors, as well as thiobencarb or bolero abolish and clincher. So we started with that, and then over time we started adding other resistance to that same biotype. So we now have added or biotypes that have added serrano resistance. And it starts to get a little bit complicated, as you can tell, to start planning your program. So it only leaves you with bute, which doesn't really work 100% on watergrass, and then propanol. So a lot of folks are already in this situation, and it starts to mean that we're relying only on propanol. And so we have started to find biotypes that are resistant to propanol. I'm not sure how widespread it is, but we can't run an herbicide program with one herbicide. So folks that are in this situation, what do? We don't have a lot of options, but one of the things that we can do is a pre-plant sale seedbed. So that's flushing the field before we plant our rice to bring the weeds up. And this works very well on watergrass. And then spraying with a non-selective herbicide like glyphosate. Or if you can switch to a drill seeded system using something like pendimethalin or Prowl is the trade name. And that also will work, but a lot of folks can't switch to a drill seeded system. So I advocate on really messy fields to use glyphosate. Foliar applications, we have the Abolish Regimen tank mix, and then this high rated regimen, which I do realize lately has not been working quite as well, and we are looking at or investigating that further as to why that is. Okay, so that's kind of a run through of our current status in terms of weed management. So what are our sort of major principles of weed management? And this applies to all crops, not just rice. So the first one is managing our weed seed bank. So that's the seeds in the soil, okay? So trying to reduce those by germinating them and killing them in some way, or leaving them in the soil long enough that they decompose. 2, preventing more seeds from being deposited into the soil seed bank. And then the last one is really knowing the critical period of competition for each weed. So that's when you need to control those weeds to really lessen the impact on yields. So here's our soil seed bank. Okay, so on the left here is our non-dormant seed bank. So those are the seeds that are going to germinate if all conditions are correct. On the right is our dormant seed bank, which means that those weed seeds are not going to germinate regardless of what happens. Okay, they've entered a period of like sleep, essentially. Okay, and even if the temperature is correct, even if the soil moisture is correct and perfect for them, they won't germinate. Okay, until something triggers that dormancy breaking, and then they will enter the non-dormant seed bank. And these seeds can go back and forth between the non-dormant and dormant seed bank, depending on our management methods. Okay, so what can we do and where can we impact things? Okay, so the first one with the non-dormant seed bank is that we can control the ones that germinate. Okay, so the ones that germinate and die, hopefully that's us doing something, whether it's cultural control or applying an herbicide. In the case of, barnyard grass, for example, that would be like flooding, right? Suppressing that barnyard grass. Germinating and live. This is not great when this happens, but this can happen, right? So when they germinate and live, because we didn't get them for some reason, then they go back into the seed bank. So they set seed, and all that seed falls back into our seed bank and continues to add to the seed bank for years in the future. Okay, we have in rice some control over decay. So we winter flood our rice fields, and that can sort of, encourage those pathogens and fungi that normally live in the soil to break down some of the seeds. It doesn't work on all of them, but it works on some of the larger seeded weeds. That flooding also can encourage predation. So birds, mice, and insects. In our case, birds are a really big part of our weed management. Ducks and geese that visit our fields love those big seeds, watergrass and rice and things like that. We also do have a bit of control over seed coming in from other locations. So seed dispersal from outside of our field into our field. And I'll talk about this a little bit more later with weedy rice. So machinery is a big way that we spread weed seed in rice, through sharing of equipment, also through planes if we're not cleaning out our planes well before we seed other fields. Even just, bank out wagons and things like that, driving around to different fields. If they've got any seeds stuck in them, a lot of that seed can have weed seeds in it. Okay. Real quick on the critical period of competition. I'm not going to cover this in great detail here, but basically it's when you need to control the weeds in order to make sure that you have the least impact on your yields. Okay, so for rice, we haven't calculated this or done the research for all of our weed species, but for watergrass, we know that it's the first 30 days. So if you don't get your watergrass in that first 30 days, it has a large impact on your yields. And the longer you wait to control that watergrass, is what it means basically, is that the bigger the impact will be on your yields. Regardless of if your field is clean at the end of the season, it doesn't matter. It's really, you got to get them when they're small during that first 30 days in order to have maximum yield. Okay, so I'll talk a little bit about some of our newer weed species in relation to this really quickly. So integrated pest management in the context of rice, weeds, really these are our mini little hammers, right? So it's basically the idea of not using just one thing to control your weeds or your pests, but many little different things. Okay, and that over time, you're gonna better manage your weeds than by just focusing on one weed management method. So we all use pesticides already, but we can use cultural controls, mechanical, sanitary. I touched on that earlier, and I'll talk about it again in a minute. We don't have biological control in rice, and we're working on some tolerant or herbicide tolerant rice varieties currently, the oxyfluorophane. But that won't be ready probably for another couple of years. Okay, so I'm going to talk mostly about my research right now in weedy rice, and then also in our new watergrass species biotype, and give everyone an update as to where we are. So weedy rice, I mentioned earlier, talking about sanitation. Okay, so sanitation for us for weedy rice is really using certified seed. So certified seed or quality assurance seed is seed that has gone through an inspection program with California Crop Improvement Association, and they have a zero tolerance for weedy rice. So, and I'll show you soon, but it really has been working on certain weedy rice types to get rid of weedy rice. The other one is hand roguing or hand pulling weedy rice from the field. And then last, we are working on herbicide use and how we can use herbicides right now, the ones that we have, available to us to manage weedy rice. So through a spot spray and a stale seed bed method. Okay, so we did a large weedy rice survey in 2020. And the purpose of this was really to both track spread to surrounding fields. We surveyed roughly 10,000 acres. We wanted to see if the fields that were infested had weedy rice moving from the fields where they were located to the fields surrounding them. So we're looking at that, and then also the effectiveness of our cultural practices like roguing and certified seed use. Okay, so as I mentioned before, we surveyed almost all of our known infested fields found since 2016, approximately 10,000 acres in total. And we surveyed all the fields around those fields. And we wanted to see the severity of the infestation. So we applied this rating scale you can see on the right side of the screen. 0 was no plants. And we did this on a basin level. Okay, so rice fields are divided into separate basins. So we did it on a basin level rather than a field level. In the past, when we had been identifying fields, we were looking at it and saying, okay, if a field has 1 plant in it, the entire field is infested. But we wanted to get a better idea of really what the infestation levels were on a smaller scale, right? So if a field is 10,000 acres and it's got 1 plant, we didn't want to say the whole field was infested. So again, on a finer scale here. So our ratings went from 0, no plants, all the way up to 6, which is more than 25% of the basin infested. Okay, so you can see that this is the acreage by county, and we surveyed most of the counties. We weren't unable to get into some of the fields in San Joaquin and Sacramento last year, but we have since updated that this year. But we surveyed most of the fields in all of the counties that we'd found weedy rice in the past. So like I said before, about 10,000, close to 11,000 acres in total. And the infested acreage was a little over 2,000 acres. So the percent infested was about 20% of the fields that we surveyed. I mentioned earlier that we have those different biotypes. So we have biotypes 1 to 7 listed here. I didn't mention 7 earlier because we're still confirming whether or not it is weedy, but we went ahead and included it in our survey. Most of the acreage is infested with type 1. Some of the acreage, or second highest, is type 2, and then type 3. For types 4, 5, 6, and 7, we found very little acreage in total. The most interesting thing on here is probably type 5. So when we began the survey, we thought we had upwards of 5,000 acres of type 5. The fields that we surveyed with type 5, we didn't find any weedy rice. And type 5 has very low dormancy. So in switching to certified seed, it actually appears as though we were able to eradicate most of the type 5 from those fields. I can't say that's for certain the only reason why, but all signs point towards that being the reason why. In terms of infestation level, again, most of the fields that we surveyed had 0 infestation. So again, positive sign. Some of our fields had infestation level 1, which was just a few plants. Some had infestation level 2, and some had 3. None of our fields surveyed last year had infestation level 6, which was the really badly infested fields. So again, most of our fields have just a few plants or a few patches of weedy rice in them. Okay, so to summarize here, so out of our rice acreage in California, which is a total of about 500,000 acres, only about 2,000 acres was found infested with weedy rice in 2020. So really very low amount of acreage. We had no spread to surrounding fields, which is again very positive. Our infested basins were reduced by about 80% on average from previous years. And that could be due to a number of things, partially surveying techniques, probably some of our eradication efforts, maybe both. We will be conducting this again in 2025, but we are also doing yearly updates. Okay, so our some of our management techniques here are weedy rice stale seedbed. Okay, so this is utilizing weed ecology so that we're able to better time our herbicide applications. So we conducted an experiment down in Davis at the UC Davis agronomy headquarters. We wanted to have a field that was far away from all commercial rice fields. So we had to establish weedy rice in the field at that experiment site. There was no weedy rice there. So we planted biotypes 1, 2, 3, and 5. This was started in 2018 with the establishment of the populations. And then we started our experiments in 2019 and continued that into 2020. So I have rotation on here. We didn't end up doing a rotation. So we're going to focus on the difference between the conventional flood and our stale seedbed. Okay, so I'll go through some of the experimental timeline here really quickly, but then I'll kind of show you a little bit more what it looked like in a moment. So for the stale seedbed, we flushed both years towards the end of May, which I realize is a bit late compared to most growers' timing for flushing or starting to plant. We flushed again roughly a week after we flushed the first time. Again, this is before we plant rice. So we're flushing to get the weedy rice up. We sprayed glyphosate a couple of days after we flushed the fields, and then we flooded up roughly 2 days after we applied the glyphosate. The continuous flood were flooded and seeded the same day as we did the stale seedbed. And that's what the weedy rice seedlings look like. So you can see there's the rice seed right there. And this is this approximate size of the seedling that we're pulling up. So we did daily emergence counts over the course of about 30 days to count how many weedy rice plants were coming up. This is our experimental setup. So we have to set up these boards in bricks in the field in order to be able to walk on the field without messing it up and disturbing the seedbed. And then we counted the weedy rice in these rings here. So this is what the water content looked like for the stale seedbed. Here on the left axis here is volumetric water content, which is just a way of saying how much water there is in the soil. So we flushed again about a week later. So you can see the water content of the soil comes up again. And then we sprayed approximately 2 days after we flushed for the second time. And the reason we flushed twice was because the soil had dried out quite a lot and we wanted to make sure we got up most of that weedy rice. Then we sprayed and then flooded about 2 days after we finished the stale seedbed. The fields were flooded and seeded with rice, just as growers flood and seed their fields. So the rice seed was pre-germinated, and we applied with a handheld backpack, but it would be the same as if you were flying it on by airplane. Okay, so looking at the emergence here, the one on the left here, the graph on the left is our flushed or stale seedbed. The one on the right is our flooded field count. So actual number of plants per square foot is on the left here. And then on the bottom is our days after watering. And each of the different lines represents a different weedy rice biotype. So we've got type 3 emerging the most plants per square foot. Same in the flooded field. Okay, but I want to draw your attention to the fact that we have plants coming up under flooded conditions. So previously it was thought that weedy rice could not emerge under flooded conditions. But as you can see on the right here, weedy rice is emerging under flooded conditions. So this is really important to know. So flooding alone is not enough to keep weedy rice from germinating in a field. However, you can see that there are approximately twice as many plants per square foot germinating under flush conditions. So flushing is going to bring up more plants. If you're conducting a stale seedbed, that's a good thing. But if you're just drill seeding and aren't wanting to have any weedy rice coming up, it's going to be a bad thing. Okay, so we want to continuously flood to suppress our weedy rice. This is year number 2, and we were not controlling the weedy rice in this flushed area here. So I'll show you guys the stale seedbed sprays soon. But the second year, because we hadn't controlled the weedy rice that first year, the seeds went into the seed bank, and then we had even more weed seeds come up the second year. So you can see the count here. The scale on the last screen was up to about 6 plants per square foot, and now this is up to 60 plants per square foot. So we had like about a 10-fold increase in the number of weedy rice plants emerging that second year. The flooded field, we had less plants emerge the second year. So I think over time, if you're flooding continuously, you should be suppressing some of your weedy rice. Here is our emergence. So this is 60%, or sorry, percentage here. Some of them, we only got about 60%, 50% emergence when we sprayed. And I'll summarize the numbers here momentarily. So not 100% control with a stale seedbed method, but still better than zero control. Okay, year 2, still not 100% control. The timing on this was based on my best guess. We have better data now, and we should be able to put together a model, hopefully in the next couple of years, to actually better time this spray application. But to summarize here, so in 2019, we sprayed 11 days after the field was flushed. Type 1, we got 25% control, type 2, 75%, type 3, 58%, type 5, nothing. So completely missed type 5 that first year. 2020, we had roughly 40% control for types 1, 3, and 5, and a little bit less control for type 2. So, a bit of variable control. We're going to reevaluate all of this using growing degree days and hopefully get a better model. So to quickly summarize here, our emergence started around 9 days. So that's, kind of giving us an idea of when we need to spray. So we got plants coming up around 9 days. Maybe we should have waited a little bit longer to get more plants to come up. Roughly same timing in 2020, 8 days. Our flooded field, we had emergence around 8 days in 2019, and emergence didn't start till 24 days in 2020. And I'm not sure if that was a temperature related thing. So again, further research. We'll be recalculating that with growing degree days. Like I said before, we have some base temperatures being calculated right now. That's Kasim Al-Khatib's graduate student that will be doing that work. Take-home message from this, though, is that about twice as many plants per square foot emerge under flush conditions compared to flooded. Okay, so if you're going to use this as a control tactic, that's something to think about. A 10-fold increase in weedy rice plants, like I said before, under flush conditions, especially type 3 and type 1. So using a stale seedbed, it can be effective to control weedy rice, but efficacy really depends on the biotype and the temperature. So if it's really hot versus really cold, it's going to make a difference in timing on when those plants are emerging. Flooding is not 100% effective at suppressing weedy rice, but it can be used as a means to reduce the number of plants that emerge. Okay, we don't know whether or not that will be reducing the weed seed bank. So seeds could be just remaining there under the flood dormant and then reemerge whenever the field is tilled. So again, it can be used as a means to suppress, but we're not sure whether it's actually reducing the seed bank. Okay, our spot spray option for weedy rice that we have right now is a biological called Suppress. So this is currently available to use in California rice. Okay, so we started our trials in 2019. We did a spot spray application in Maxwell. The weedy rice stage was late boot. So the plants were very close to heading. We applied our suppressed product with a handheld backpack sprayer, and we held it about 12 inches above the rice. We did 5 plants per treatment this year, and this was on weedy rice biotype 3 alone. And we did 2 timings of suppress, which sort of made it 2 different rates basically. So held it for 5 seconds, held it for 10 seconds instead of remixing the backpack. Okay, let's take a look at our control here. So with that lower rate, or the 5-second timing, we had really good foliage control, 90%. Okay, same thing with the 10-second timing, good foliage control. The weedy rice panicle control, however, was quite poor. So because we had sprayed so close to heading, most of the panicles still emerged from the boot, and we ended up with this situation. So it's a little bit hard to see the panicles, but these sort of like fluffy looking things here are the rice panicles that have emerged from the boot and are still viable. Okay, so 1 week after spraying, the plants had recovered, and so this timing is too late. You can see though that the foliage looks very, very well controlled. So we did another trial in 2020. This time we did it at the early boot stage, and we did this in Davis at that field that I was telling you about earlier. Same application method. We had 2 fields this time. We had 10 plants per treatment in one field and 4 to 5 plants per treatment in the other field. This time we had weedy rice biotypes 1, 2, 3, and 5, but unfortunately it's impossible to ID these before heading, so we don't know which type was which one we sprayed in the field. We did a little bit lower rates this time, 3 and 5 seconds per plant. But our control was much better. Okay, so we also this time looked at our damage radius to our rice plants surrounding the weedy rice. So about 1.5 feet both times, regardless of duration of spray there. The foliage control was good, 97, 99%. And at 7 days after treatment, in field 2, we had a little bit less of a damage radius, but still, roughly the same as field 1, still really good control. You can see what it looked like 1 week after spraying. So plants seem to have completely died, and I did go back at the end of the season to just make sure that nothing had headed, and no, nothing had headed. And I did have one grower use suppress in the field last year, and it worked. It did work very well in the field as well. To just make sure that it works on all of the weedy rice biotypes, we also did a greenhouse trial. We did early boot and late boot, and we applied it in the field with a handheld backpack sprayer. Same deal. We couldn't get it in the spray chamber because the plants were too large. But this time we tested each of the different biotypes individually to make sure that it worked. And we also included M206, which is one of our most widely planted rice varieties. It was 100% control, both at the early and late timing. Again, this is individual plants. So this is our untreated versus that earlier timing and the later timing. So really, really clear, good control. Okay, some reminders here. So it is currently available to be used in weedy rice spot spraying. The label does not allow for application when there is standing water in the field. So this is really important to note. So you have to drain the field in order to comply with the label. And the label is the law. You need to drain your field before application. And the timing that we recommend right now is after the last grass herbicide has been applied, but before the weedy rice has started to flower. So usually about 60 days after seeding. If you go much earlier than that, you might miss some of the weedy rice. Reflooding is recommended within 48 hours. And the reason we recommend that is because of additional weed germination. Again, follow the label rate. Please don't go off label, even for spot spraying. The maximum label rate is 9% solution and 100 gallons of water per acre. And we're working on maybe on a label amendment to accommodate higher rates, but I don't know if and when that will happen. So for now, just follow the label, please. Further research, I would like to try suppress in lieu of glyphosate. I think it would work, but this way we would have a biological that is registered for organic rice as well. Okay, so moving on to our new watergrass species or biotype. What herbicides are still usable and useful? And it's also very necessary to better identify these species and biotypes. And I'm currently looking at whether there's a correlation between efficacy of some of our herbicides and the species and biotypes that we have. So I did a preliminary watergrass herbicide screening in 2020. We collected populations from 10 fields in 2018. We had first identified this particular species or biotype in 2017. So again, we collected more populations in 2018 and then did this screening. And this again is just preliminary, still working on doing a larger screening. And we collected 60 some odd samples last year, 64 samples, and we're currently conducting a screening right now. So this is just preliminary, but we're doing a more comprehensive one currently. So we used all of the currently registered herbicides that are supposed to control grasses in California. Again, here's the active ingredients, and we used field rates. I didn't go above the label rate. I didn't do 2x rates or anything. And in some cases, I didn't do the maximum label rate. I basically did what sort of is the normal field rate that folks are using. Looking at percent control. So this is a sort of a heat map here of our different biotypes. So here on the left are the different biotypes from different counties and our 2 susceptible populations. And this is percent control compared to our untreated control by fresh biomass. Okay, this is roughly 2 days after treatment. These were all treated in a spray chamber in the greenhouse. So when the plants were quite small. Okay, so just real quick, we won't go over this in great detail, but you can kind of see the darker colors means that there was no control, or the red colors. The greener colors means that there was good control. So generally speaking, we did not get good control with any of our granular herbicides. So Granite, Serrano, Bolero, Butte, we did not get good control. Okay, propanil, regiment, and clincher, better control. Okay, the only caveat that I would say to this is that the plants were quite small. This is maybe not when we would be applying these herbicides in the field. So a bit early compared to normal applications if you're using it as a follow-up application. This is percent control in terms of number of living plants. Again, the number of plants that survived the application. So if it was 0, that means that 100% of the plants survived the application, so poor control. And if it was close to 100 or green, that means that none of the plants survived. So again, really looks like propanil and clincher are probably our best bets. So with the regiment, I am not sure if all the plants that were treated would survive. They were small, but they may not survive and produce seed. So we need to do some more of this or follow-up studies in the field. Okay, so last part here of the talk. So this is where we start to talk about how hard it is to identify weed or Echinocloa. So we have, as I mentioned early on in the talk, 3 known echinocloa species in California. This one here to the far left is barnyard grass. The one in the middle, late water grass. The one on the right is early water grass. Same species here on the right, and the photo on the right. First one is late water grass. The one in the middle is barnyard grass, and the one on the right is early water grass. Again, those are our known echinocloa species. So watergrass, just to back up a bit here, it is known to be difficult to identify. Worldwide, weed scientists and botanists have a difficult time with this particular family of weeds. Most of the distinguishing characteristics in the botany manuals and things have to do with seed size and other seed characteristics. So for us, that's really too late to be able to ID these plants. At this point in time, we're unsure if we have a new species or if we have perhaps a new biotype. So it could be a new species moving into rice. We might have something that was in another cropping system and it's moved into rice, or we could have something from another state, unsure, or we might have a new biotype. So we're currently working on this at the UC Herbarium, UC Davis Herbarium. So a closer look here. So on the left, again, late water grass. In the middle is barnyard grass. And on the right is early water grass. Again, these are our 3 known species. This one here that I showed early on is the unknown species or biotype, which right now we don't have a name for, but we're referring to it basically with these, or identifying it with these long purplish awns. So to summarize really quickly, what we're utilizing right now for description and identifying. And again, this is not good because it's the seeds. It has nothing to do with the plants. So when they set seed, it's too late. So our new biotype or species, we're saying has smallish seeds. Okay, so smaller than late water grass and early water grass, and has those long reddish awns. And again, those awns are the thin little things that stick off the end of the seeds. Jungle rice, which I didn't mention, is pretty much just found around the edges of our fields. So it might be confusing if you're out looking for weed seeds. It has extra small seeds, so very, very tiny seeds, and the seeds are not awned. Barnyard grass, the one I mentioned several times, has smallish seeds, and it has variable awns, which means that it might have awns on the panicle, on like different awns on one panicle. It could have some panicles with awns and some panicles without awns on the same plant. Late water grass has large seeds with no awns, and early water grass has large seeds and their panicles are completely awned. So when you compare that to barnyard grass, I said before, has variable awns. Early water grass is completely awned. So we currently have some work in progress to hopefully help us with the identification. So we're measuring phenotypic data. That basically means like how the plants look from the outside characteristics, as opposed to genotypic if we did a genetic analysis. The data includes height, tillering, number of panicles, date of heading, on length, color, et cetera, et cetera, et cetera. So we're measuring, I think, roughly 20, 30 different characteristics on these plants. We've sampled them all, and they're all pressed at the UC Davis Herbarium right now. And we're working with our herbarium curator to hopefully help us identify them. And the hope is that we might be able to develop a new key for California, at least for California rice, that would help us to identify these species. So again, it's kind of early on, but we're working on it. Okay, so to summarize, weed management in rice is only going to become more and more complicated, right? There's fewer herbicides available, and it's difficult to register new herbicides for a variety of reasons. The fact that there's not that many new chemicals coming down the pipeline, on top of the fact that it's just difficult to register things now. It takes a long time. So what can we do? So we really need to preserve the tools that we have. We have new herbicides coming down the pipeline, and we need to steward those herbicides. So that means following label instructions, making sure that we're applying at the correct label rates, that we're, not doing things that we shouldn't be doing with those herbicides. Use multiple methods of management. That means using integrated management tools. Some of the things that I've talked about: stale seedbed, perhaps sanitation methods, things that we don't normally think of maybe as weed management tools, but using those and in collaboration and cooperation with our herbicides. Rotating our herbicide chemistries. And again, I mentioned earlier that this is pretty difficult to do in some places, in some fields, but trying to do our best to do that, especially when we have new chemicals. And then lastly, if you notice a problem, call someone. So our new weed species that we found all of them because PCAs or growers called us and let us know that they had something weird in their fields. So make sure that you're calling your advisors or ag biologists in the area to let people know if you think you have something, because if we don't know it's there, it can really quickly take off and take over a lot of area. So look out for new weed species and let us know. And then if you have herbicide efficacy issues, please, again, give us a call. We have a resistance testing program with UC run by Kasim Al-Khatib, and he does that for free for growers and PCAs and California rice. Okay, and I think I basically talked all the way to the end, but I have a few minutes and I can hang on for questions.
We have a few questions here in the Q&A. The first one is from Daniel. He's asking, is rice still being planted on rows instead of flooding?
So in California, we mostly plant in a continuously flooded system. So most of us do not plant in rows. There's probably, I want to say roughly 2% of the acreage, 2-3% of the acreage is drill seeded and planted in rows, except for in the Delta. So that if you're down in the Delta area, San Joaquin Valley, they do plant it only drill seeded down there. But the rest of the acreage, which is the majority of the acreage, is mostly flooded.
And then the next question is to control just the resistant grasses, not weedy rice. Is it still necessary to flood for 11 to 15 days, or is a few days long enough for germination?
Oh, that's a good question. So for most of the grasses, it seems to take roughly, I would say 7 to 14 days to get most of the grass up. Although that does depend somewhat on temperature. So if it's warm, it will come up faster. So, we have some research on it, but it's not the timing isn't perfect. But I would say probably roughly 2 weeks you'd get most of it up. But if you went roughly 7 to 10 days, I would think you would get probably 80% of the grass.
Okay, thank you. We have one more question in the Q&A. The question is, can you please explain what the 3 seconds and 5 seconds mean?
Oh yes, sorry about that. So that was for the suppressed timing. So that basically meant that we held the sprayer for 3 seconds. So counted 1, 2, 3, and then for the other one, 1, 2, 3, 4, 5. So that was how we did the timing instead of remixing the backpack twice. And we thought that was a better or easier way to sort of simulate what somebody would be doing in the field. Because it's a handheld backpack sprayer with like one wand.
Okay. All right, so that's all the questions that we have. It looks like we're all ready to head out. Thank you, Whitney, so much for speaking today, for bringing us this information. And thank you to everyone who attended, and have a great day.
Thank you all.